Well, I can introduce myself. I'm Andrew Cinderella. I'm the process manager of R&D at Purisys. We're a small CDMO and generic API producer. We're located in Athens, Georgia. One of our kind of niche fields is that we have controlled substance licenses for schedules 1 through 5 in the DEA. So we specialize on our CDMO arm of helping customers who have potential investigative APIs that will be controlled substances or come from controlled substances, and help them handle those and manage their way through the DEA maze in how to get them at least to the clinic and then all the way through to commercialization if their APIs are worthy in the clinic. And then we focus a lot on small volume controlled substance generic APIs for smaller patient populations. I've been in the industry working in CDMOs my entire career for a little over 8 years now.
Yeah, hi everybody. Myrna Monck. I'm from BPFX Solutions. Prior to that, I worked for 25 years in GSK, principally in drug product, and most recently in the sterile division, looking at scale up of all modalities, but principally with peptides, oligos, and biologics. And so from our perspective, really the cost quality time curve was very important in terms of understanding what that scale up timeline and capacity looked like in order to meet the demand of the clinic and then eventually the market.
Any initial questions? No? We focus a lot on small molecule scale up. So some challenges I would say that we've seen in the recent few years, and I would definitely say across my career, is an increasing rigor in the earlier stages of development from customers and innovators. Is they're seeking at early and earlier stages to have more and more rigor closer towards later phases, so higher purities, earlier identification of impurities and risks, especially with nitrosamines. Guidance coming out is people are asking earlier and earlier in their drug substance programs about nitrosamines and genotoxic impurities. And so normally, earlier on, I would say in my career doing process development, most companies were mostly worried about let's make it the thing, and now they have a much more risk-based approach that they want to take. And so what I would say is they're taking a lot of quality by design tips that come in the technical side of process development and taking it and applying it to their risk evaluations for their small molecules, going for the end game of approval and trying to design that quality up front for risk assessment of nitrosamines and potentially mutagenic impurities.
Yeah, and on the drug product side, particularly for oligonucleotides in the large biofarms, cost is a big factor because they're very expensive. The APIs are expensive, and so the risk evaluation takes precedence. What size batches are we going to manufacture? And if it goes down for a sterile batch, you're taking tens of millions of dollars of risk if the batch goes down effectively. My experience has been this gets scaled up in multiple stages. We start with meeting the demand for the early stages of, let's say, the market, where you have an S-shaped curve, and then scale up multiple times. So you could start with, a 50-liter batch to supply the market for roughly an orphan indication, depending on the overall dose and volume. And then scaling up to 100-liter batches, 1,000-liter batches, and beyond, recognizing that for a sterile batch, there's always the risk of lack of sterility, and then the entire batch is scrapped. So that's one of the key factors that that factors into the scale for drug product is really the cost of the batch as well as the risk of the batch going down due to factors that sometimes are beyond the control of the manufacturer themselves.
Got a question over here.
So a question for Andrew. So we're talking about companies looking at, say, GTI risks and nitrosamine risks. Certainly, I just recently retired from Sanofi, and we were certainly doing that. But did you see that from, say, startup biotechs that sort of have their first product going into early phase clinic, where maybe they're just looking to sell that once they get some clinical data? Are they doing that to have that as part of the package to sell, or are they not? They just want to go fast and not really worry about it. So what are you seeing there?
Yeah, so we do have quite a number of virtual biotech startups that come through our doors, and they are considering those things. And some, the experience level of the people who work at those companies varies quite a bit. And what we see is, depending on their level of experience, it tends to be that those who are less familiar with some of those topics are more risk averse because they're not sure, and they lean on us as their CDMO to really try to help advise them on how much they should look into it. But they are starting to consider that to go into their packages, that they've considered it. And I think part of that is, for some of these virtual biotech startups, they want to be mature and appear mature, especially if their goal is to sell their molecules right. They don't want to seem like they're just a company who is going right off the rip and just trying to get clinical data and jettison a molecule. They want to show that they have an understanding of what they're doing. And that's something that we've spent a lot of time as we do have some of our own generic APIs and have expertise in doing mutagenic impurity assessment, nitrosamine assessments, as well as nitrosamine testing and NDSRI synthesis. We help advise them to the best of our ability on what we see and what we kind of collectively we hear from a lot of our customers, but they are thinking about that. Yep.
So my question is for the scale-up challenge in peptides in the final API isolation. Right now, people use the precipitation or spray drying technology to address the lyol bottleneck. So my question is for the spray drying technology. What kind of status of this technology to be applied in peptide API? And precipitation more mature, and how we look at the spray drying technology.
So at Purisys and most of my career, I've focused a lot on small molecules. I don't have a lot of hands-on frontline peptide experience. I think that's why we were bringing on a couple of our other panel members who aren't here, unfortunately. I'm not sure where they are. But I have heard that, I can speak a little for small molecules when it comes to things that are water-based that you need, obviously lyophilization is kind of, I don't know if I'd want to say antiquated technology, but we don't like it. It's slow, batch to batch. Spray drying really is one of the ways people are moving for those kinds of compounds. And you really just got to be careful about kind of molecule stability, and I'm sure that applies to peptides as well. If you're using spray dryers or spray drying with like heat or anything like that, and how that's impacting morphology as well. Mm-hmm. All right. Any other questions?
Any other questions? Yeah.
Okay. Well, we can just sort of riff on it then and talk. So one of the things that we really see is we have seen, obviously, across the industry, AI is a big buzzword. So we've heard a lot of companies asking, and that's one of the things that they tend to want to ask us when they come and assess us is they don't only want to know what's your equipment, they want to know what kind of analytical tools you're using to analyze data. They don't necessarily want traditional just statistical DOE anymore. People are asking about machine learning tools and AI tools. That's something that we've started to look into. But across the industry, chemistry can be complicated, and even with small molecule APIs. And so where the forefront of AI technology is, chemistry technology and chemistry tools are not there at the forefront of AI technology, because if you saw any of the presentations from some of the companies here, such as I think Chemlex is here, they're like half AI company and half chemistry company, and not every company has those kinds of in-house expertise. So one of the things we look towards is kind of some out-of-the-box platform tools that we can contract with, such as software as a service platforms. And we're starting to learn some of the ins and outs of that and how that's beneficial and where we could find some pitfalls. But we have started to see that those are some useful technologies and can be helpful for scale-up if we're able to input some of the right parameters to help with that.
So for the scale-up of principally, I'll talk about oligonucleotides. One of the key operations is rehydration, and the rehydration of oligonucleotides is long. They act like hydrogels or polymers, so they swell before they go into solution. It takes a long time before you actually get this completely solubilized. And for each of the sterile manufacturing steps, particularly before the first filtration, there are hold time requirements. So for oligonucleotides, one of the things that we explored was, could the oligonucleotides, for example, the final step in the API manufacturing step, be a rehydration step or a TFF step, in order to have a frozen, basically a frozen liquid containing the API. So essentially the oligonucleotide wouldn't have to be rehydrated. This actually reduces the risk for the manufacturer of the oligo in that manner. If there is bacterial ingress, higher level of endotoxin prior to that first filtration step, you end up having to turf the batch if you don't meet your endotoxin specs, and that ends up being very expensive. So an evaluation of having not a spray drying or not a lyophilization step, but having an API that's in a frozen liquid is an advantage for further processing. For, you mean the final API? Yes. So, so your QC methods will be already in the solution state.
Take it one step further. Is that very similar to the concept of taking into bulk formulation? So instead of isolating behind, isolating, essentially going one step further to bulk formulation, which you can take into a product. That's what we call your.
Exactly. So that's so if I can make an analogy with biologics, for example, the formulation step essentially is the final step of the downstream process. It's TFF, ultrafiltration, diafiltration, and the final steps prior to the filtration into the primary container is a TFF or a formulation step. This gives you an advantage from from downstream processing, where you don't have to worry about that. You already have some sort of bioburden reduction at that point.
Sorry to monopolize, but is there like a regulatory challenge of doing that? So is there a regulatory challenge with the FDA in calling that your isolation step versus an isolation step of pure?
It's a good question. I don't know if we have any regulatory folks here, but from that perspective, there would be a discussion with the agency in order to characterize and classify that as a different step. Yeah. Okay.
So I'm pretty comfortable with small molecules, but I'm trying to understand these large biologics in terms of the separation portion. So purification and separation. Maybe it's a concept of volumes of something like it. But is that a big challenge? Are we just essentially scaling up lab separation still? Or are we changing techniques in purification and separation?
There are differences. In the biologics where you're looking at bioreactors to create the biologics, you get a lot of cellular components and debris that you have to separate out. There's a lot of host cell proteins, host cell DNA, and so the steps are refined accordingly. The real, I guess the biggest or the most expensive, certainly, purification step is a protein A column. The column runs millions of dollars because it's essentially a protein attached to a resin. It binds to the tail of the monoclonal antibody, for example, or other. And most of the rest of the constituents actually come through the column, and then there's an elution step. And that essentially is the main purification step, and that's followed by several polishing steps and viral reduction steps, viral inactivation steps associated with that. So purification also has to make sure that your protein is going to be intact by the time you get it into formulation and solution. So it's largely aqueous.
So I'll let them ask them all the questions. To your question on the AI, just it has to do with regulatory again. But the question is, I can understand the idea of using machine models essentially to gather information. Okay, where are we using, or is the industry actually using them in any way where it actually impacts the process? And if that's the case, how are we making sure that the validation portion is done and you know, and it's consistent and reproducible, let's say it this way, because you don't have to backtrack validation all the way into the machine languages. So I understand the question. In other words, I can understand using AI to say, hey, find me a good synthesis route. Okay, well, that's early discovery, you know, it's early
Yeah.
route scouting and that sort of stuff. But my question really is anyone using it towards the very end? In other words, look at this, the analysis that's coming out of this batch. Now control this batch, you know, and change my feed rates or temperature control or whatever the case is to be. And if that's the case, then it becomes a control mechanism which is now needs to be validated.
Yeah. So we don't use it as a late stage. I'm sure some people are investigating that. One of the big things is I believe there was a FDA release, though, that it's really important if you're using AI tools that they have transparency on how decisions are being made. So what they don't want is essentially you to say, well, the AI told me to. That's really bad. I mean, it's the same as if you have a chemist come in and he just says, well, I think this is better, and they don't have any data. So that's actually one of the challenges, I think, with AI, and machine learning is you need to have some level of transparency on how decisions are being made. And you could go through and just say, well, we proved that the decision is right, but they still want to know how. And that's one of the important things that they're looking at, and why obviously process chemistry is so important, because they're still concerned about the process and how those decisions are made. And that can be a challenge, is my understanding for the AIs, is getting them to give you the rationale for how they reach their conclusions in a way that we can understand, without necessarily, I guess, being an AI expert.
I guess in the drug product space, I don't really know if people are using AI in a similar manner. They're embarking on an awful lot on documentation, regulatory filing, using templates and AI tools for regulatory documentation in particular. And really that's a time and effort saving approach, where you start with a prototype and you effectively use several templates in order to craft the first version of a regulatory document, and then you go from there. And this spans the entire gamut across drug substance, drug product, analytical, and so on. But from the perspective of the science and scale up engineering, sterile manufacturing, people are looking into employing things like digital twins and evaluation of the unit operations and the processes in order to better understand the risk, the process, the duration, timing, and so on.
Yeah, and it is a buzzword, and if you went to any of the other, I think it's the first day we had the AI workshop, they talked a lot about these systems are based on learning, and so you have to have a lot of data, but your data has to be of high quality, or you'll have models and systems that don't work very well. And that could be a challenge in itself if you're not already an organization that focuses solely on high data collection, per experiment, if you're not collecting the right data, or understanding your processes well enough to input the right data into the system. You could collect tons of data, temperature profiles, heating things like that, impurities, but if maybe you're not using the right inputs into your system, you're just going to get garbage out. And so there's a learning curve. And there's also a challenge, right? As I said, the cutting edge of AI technology and then the chemical industry and the pharmaceutical industry are not necessarily matched there. And I think some of that also comes from, there are a lot of chemists out there who has tons of experience, and maybe they aren't quite as skilled in computer technologies. And so taking workforces that have sometimes been very steeped in, say, traditional ways of doing things, it can be hard to change ways. So that's one of the challenges, is just people. And I mean, that's probably a challenge throughout history of new technologies, is getting people on board who've never used them before, or maybe are skeptical of them, and really getting them to embrace these technologies. That can be a challenge and a barrier to really pushing even just small molecule chemistry into kind of the next era in the age.
So, have you seen any uses for AI or these new technologies that are coming out beyond data analytics? I mean, it doesn't sound like there's much help, because I feel like another aspect of like training an AI model that maybe isn't always in circulation of discussion is, how do you control that IP?
Yeah, that is, in a couple companies I've talked to, yes, that is a big concern, is people are worried that if they put their information into an AI system that they don't own, what is that company going to do with it? And a lot of companies will say, we keep things separate. And, if you're, a chemist, you sort of have to trust them. If your background is chemistry and managing chemistry, you have to trust them. You don't have an IT background. Hopefully you have an in-house IT that can at least help you assess that. Like, is that true? Are you asking, can they ask the right questions to understand that? I've heard that question asked a lot about who owns processes or IP around when an AI comes up with something. And my understanding is that's a tangled legal question. Obviously from the side of companies, they want to say, well, it's ours and, we put in the information, so whatever comes out, it's just a tool and we are the IP owners. I've seen AI companies will say, well, our AI is the owner and we're going to put the AI on the patent. And patents are really mostly, from my understanding, right, they're made for people to be the owners and the assignees of, and companies, but not necessarily tools and computers. So I think there's overlap there with legal, entanglements that I'm not really as familiar with to talk about. But I mean, obviously there are AI systems that people are using to do protein and small molecule docking in silico to check those things, to try and do drug discovery with AI. And that's based on some input data from maybe known ligands or known results. And then they go and say, hey, here's the binding pocket, here's a few things and their binding affinities. Help me find something new. And then what they're doing is really looking for some innovative new molecule that it's going to say, hey, why don't you go try this one out? And then making that, yeah.
From the discovery perspective, I've seen a lot of consortia formed around a number of different players who, I don't know how they sign on to the legal aspects of the consortia for AI, but there's a lot of sharing of information in order to push discovery along in particular. How that moves into the development space, I don't know at this point.
So both from a drug product and drug substance perspective, can you talk a little bit about where you think continuous processing is going in the future, as well as, or at least in chemistry aspects of flow systems and things like that? So talk a little bit about how you see the future on both sides of the continuous piece and where the advantages and disadvantages.
Yeah. It's continuous manufacturing, full manufacturing is not something we do, but I am familiar with those concepts. And it is, I think, somewhere that a lot of companies are going and heading, the continuous manufacturing rather than batch to batch, designing systems that can help you avoid high volume batch to batch limiting steps and using surge tanks and the combinations of constant continuous crystallizers to help increase batch sizes, to help reduce QC and QA batch releasing. So if you can just double your batch size, just one time, you can reduce testing burden by half. And a lot of times what we see and what companies will say is, that's great, we made it, how long does it take to release? And so if we can have less releases, we can help increase, product out the door. And then I would also say for a lot of steps, we see things like typical flow chemistry and continuous manufacturing problem solving, such as impurity formation, because you need longer reaction times in bulk and in batch, and then you have impurity formation because you're trying to squeeze out, your last 10% of conversion. And so I think those types of systems and engineering can really help to perform reactions quicker and help reduce impurity formations and just help the efficiency of your overall process. So I think that a lot of companies are headed in that direction.
Yeah, from a drug product perspective, a few things come to mind. How do you define a batch? And how do you release a batch? And early on when GMPs for the 21st century were introduced, we embarked on a process for identifying, I would say, real-time release of processes. And this was in the early days, and this is just to give an example. So the models were generated on loads of data for a marketed product. It was sent to the agency. There were lots of discussions, and then the agency, this was with the FDA at the time, they basically said, we're not ready for you. So I think the lessons learned from that really were you engage with the agency, but also with your quality representatives in the organization in order to be able to understand what do you release on. If you have a model, what are the parameters, the values that you release on? What does the specification look like? So it's the definition of all of those touch points that are going to enable, I think, what's defined as a continuously made batch.
Mm-hmm.
So do you think those are specific for drug product? Because Genzyme was making Renagel, Renabella by continuous process with the API, and releasing based on, basically produce a material so long, collect that material, you say this is a batch, you do your analytics on that, even though you've had in-process controls the whole time. So do you think it's more of an issue for a drug product than for a drug substance?
We'll be in the sterile space.
Oh, sterile space.
Yeah.
I can see that, yeah. And then just, if you don't mind asking, so going back to, so I've been interested in flow chemistry for a long time, and the problem was, well, everybody has all these reactors, and so why we want to invest, right?
Mm-hmm.
And then there was some reason to invest because, well, you could do, say, electrochemistry for redox, or you can do photochemistry and chemistry that you either can't do or it's much harder to do in batch, or there's high reactive stuff that you don't have to do it in the desert in case it blows up, right? You can do it in normal places, which drives some things. But do you still feel that, in your experience, there's, unless there's a real good reason to invest in flow, why do it? Because we have all this other kit already.
Yeah, inertia can be strong, right? If you've already got assets, there is always a tendency to say, well, just keep getting money out of the assets that we already have. We've already paid for them. They're already paid off. And then, what is, the, business opportunity for these new systems? Because they are expensive to implement a brand new flow chemistry setup or a brand new engineering of continuous manufacturing. It can have a large startup cost, and innovators have they have blockbuster drugs and they have lots of revenue, and for CDMOs that aren't in the business of trying to market anything for themselves, they are watching their margins and they're watching those business opportunities. So I think there is still places for batch chemistry, and particularly in the early phases, it may not be as beneficial to spend months of time trying to optimize a flow or continuous manufacturing regime when you could knock out a couple batches of clinical material and be done with it in the same or less time. So I think batch still has its place, and especially in historical legacy processes at companies, it's probably not as worthwhile for compounds they've been making for decades, especially with validated processes, and then the regulatory impact of changing processes once they're validated, especially older processes, then you're going to reopen or have to file brand new DMFs with the agencies and things like that. So I think it's sort of coming along, and hopefully as products progress towards the later stage, that starts to become something that more CDMOs have the capability to start to look at and say, here's sort of your line. If we keep going further, it might not make sense to go back and retool your process for a change to continuous or flow, but here's a spot where you see promising results. Perhaps now is the time where we talk about, is it worth the money to move to a flow or continuous manufacturing regime for you?
I would argue it's never a good time. And that's the problem, especially in pharma. You do it, okay, let's do some stuff for a sample prep, then all of a sudden you have a process that's locked in. Now you have to be very nervous about changing the process,
Mm-hmm.
which will happen when you go to flow.
Yeah.
And you never want to do it. And so you start scaling up and you get locked in. So the only time it makes sense in my mind is when generics come in.
Mhm.
Generics should go down the line of flow because they have an optimization step there
Mhm.
where they can do it, but it doesn't help with developer.
Hmm. Yeah. Yeah, I see that. That's a pretty good take on it. Yeah, your generic is already, if you can get it made, right, it's approved, you can get it to market, so the money is there.
You have your time to develop a different process.
Mm-hmm.
That's my point. Well, if you're a brand or the initiator, you just don't want to touch a change. You just don't want to make a change. So
Mhm.
you either have a conscious choice. You say, okay, look, when we scale this up, or after whatever, phase 2 or something like that, that's when we're going to do it. It's very hard to change.
Yeah, I mean, if you have a challenge, so you have a challenging molecule and you just went and said maybe it's the Band-Aid on the medchem process to get it into early clinic, and then say before 2B, you switch to a flow process, and then you're locked
That's
the
what
rest
you
of the way. You can do that.
Mhm.
You almost have to design it.
Mhm.
So I can say in the drug product space, one of the areas of interest has really been not in scale up, but in scale out. So with some of the new isolator technologies that are small, if you could have replicates that gave you essentially the same output, and there are ways to define that, then you can have multiples of these in a large facility and run 1, 2, 3, 10, 20 to actually define or generate as much material as you need for your demand. And that makes an awful lot of sense when an individual batch costs so much. So if you can manufacture 5,000 to 10,000 units off of a machine and do that across in a parallel fashion, it gives you much more of an advantage in terms of cost and risk.
Any additional questions?
Well, I have one thing. How much is sustainability factoring into now scale? So, I think we're sort of on the tail end of where this used to be a big thing and everybody was talking about it and then reality hit and, but realistically, how much is it now? I mean, how much is it really considered? What solvents, how much solvents and all that sort of stuff?
I would say we haven't really seen customers come in and tell us that green chemistry principles are a priority for them, to be honest, especially not in their early phase work for sure. They just want to get it made, and volumes of solvent are usually limited to being driven by just process efficiency and throughput. They're not really asking us about solvent recycling and things like that. It's very similar. They talked about it, I think, in one of the first panels for peptides. A lot of people's main goal is to push their projects through, and I think it was a big thing. And now a lot of the green chemistry principles are sort of taking a secondary backseat to just trying to get projects pushed forward. At least that's what we're seeing.
I can add a comment on the drug product side. In terms of sustainability, it really represents about 5% of the opportunity, but there is a huge push, for example, in plastics recycling, particularly in Europe. There are a lot of requirements for having at least 30% recyclables, or it might even be higher compared to the last time I looked. So, so there is a huge push depending on who you're asking, what the market is, and where you're actually doing the manufacturing.
Affecting the packaging.
It is. It's very much affecting the packaging because we're talking a lot of plastics and glass on the secondary side, yeah.
So I can say that when I was at Sanofi, which was less than a year and a half ago, at least internally, we did a lot of, as we evaluated processes, certainly looked at all those sort of green things, as well as projected cost of goods and number of steps and all that kind of stuff. So it was still, at least within Sanofi internally, an important criterion on how we selected our processes. But then when we go to a CDMO, we pretty much have a process that usually we have a process that we want them to do at least for later stage stuff. I think that's, anyways, that's Sanofi, one example in big pharma.
All right. Any other questions? Topics?
Okay.
Concerns?
So just a question on some of the injectable stuff that you're doing. So how much of it is with how important, I guess, is a fancy device or something for, say, market penetration or whatever in a competitive market?
Well, it really depends on the market that you're going into and the commercial evaluation. Early on in the clinical stages, and even for, if it's an unmet medical need, then essentially it's not that important. You do have to have some sort of a safety device on top of the needle, so in order to avoid needle sticks. That's the minimum. Where you're going into a market which has a lot of devices already, then that's very important for your first file and launch. And then there's something in between. You want to ensure if, for example, it's self-administered, that the patient is going to be able to administer it, and so there's a lot of human factors issues there. So, so the answer really depends on what you're going into, the size of the market penetration. Is it already established, and eventually, do you get there with an auto-injector device? That seems to have been the trend.
Yep.
As the market goes on for small molecules, we see the molecules actually pushing closer and closer to becoming larger. I was curious if you've seen any creative solutions to solubilizing or dealing with both at drug substance and drug product stage with these difficult to put in solution molecules.
Yeah. Yeah, it is interesting. So we've seen it kind of across the board. We have seen customers having larger and larger molecules, but we've also seen some new spaces where people still are exploring some of the smaller molecules, but some of the complexity of the molecules has definitely increased. Newer functional groups that aren't as typical and that do have differing solubility factors. So solubility screening is, of course, kind of the traditional way. But I know in some cases we've looked at things like additives for solubility, so like adding certain salts or acids and bases to try and get beyond just pure solvents or just binary solvent systems to get them solubilized. Mainly for us for processing and analytical uses. We've run into some of those issues. And we have run into seeing some more zwitterion type small molecules, and those can pose solubility problems as well. And each one of them is rather unique in which solvents they have affinities for, and we've seen synergistic effects in binary solvent systems, right? So they're greater than the sum. So it does sometimes take some creativity for some of those lesser soluble compounds.
So in my experience, we've dealt with some reasonably greater molecules that will bring two proteins together to degrade, and they're like 800 microgram kind of thing, so they're fully soluble. And what we've gone to is stabilize amorphous, so make an amorphous material, add a polymer and spray dry it, and so that's kind of where we've gone. There is things on the market that are formulated that way. But it's I go back to my medicinal chemist and can't you give me a little bit more solubility? And they say, well, no, you guys have to fix it. I can't fix the toxicity, I can't fix the potency, but you guys can fix the solubility, right? So,
Mm-hmm.
but anyways, that's kind of where we've been at.
Mm-hmm.
Not happy to go there, but
Yeah.
and, people talked about co-crystals and things like that, but I haven't heard of at least when I've talked to folks, it's been kind of more trial and error, and hope you're lucky. But I think it's a tough road, and I'm not sure. I don't know if anybody else in the audience knows about how successful that approach has been, the co-crystal approach, not a salt, but a co-crystal.
Yeah.
I don't
So-
think a lot.
Well, one of the things that I've seen, for example, we've dealt with peptides, for example, which have been quite hydrophobic. So you add charged residues in there, and as long as you don't interrupt the business end of the molecule, the binding site, then you can increase the solubility by strategically placing charged residues. So what people have done is they've started with the parent, and then they've gone back and say, okay, so this is soluble in DMSO and nothing else, essentially. And then you go and then you go back and start adding some charged residues, and that's where you see things coming into the solution.
Are there any other questions to wrap up? I think we are getting close to our time. So, okay. Well, thank you to our participants.
Yeah.
And in the face of missing two of our panel members,
Yeah.
thank you all.
Thank you.